So, our video for today is all about how cells make more cells. That is a cell's second job, is to be able to reproduce themselves and make more cells. So, when we're talking about cell division, which is how cells make more copies of themselves, we call that process of cell division mitosis. And before we get into mitosis, we have a phase called interphase. Interphase is basically the group term we have for the rest of the cell cycle besides mitosis. So this is G1, S, and G2. So it's just kind of our catch-all term for those things. And it was developed before we knew what was going on in interphase. And so oftentimes it gets called the resting phase because when we were first looking at cells, cell division was easy to see, but what was going on inside the cells between cell division was not as easy to see. So do you agree that interphase is actually a resting phase or not? Something to consider for your notes. Okay, so let's talk about the first phase of mitosis, the first step in cell division. The first step in cell division, or mitosis, is called prophase. The things that happen in prophase, the chromosomes condense, the nuclear membrane or the nucleus disappears, and centrioles start building spindle fibers. These are centrioles right here. They are a type of organelle that makes the cytoskeleton, and they are really active during cell division. Now, let's talk a little bit about chromosomes. So we said during prophase, chromosomes condense. So what are they? Chromosomes are just DNA that have been wrapped around some proteins a ton of times. So if we take a DNA double helix, that ladder that we talked about before, and we wrap it around some proteins called histones, and then we coil up those strands into a spiral, and then we take that spiral and then spiral it up, that is how we get a chromosome. A chromosome has several parts. A chromatid is half of a chromosome. A telomere is the end of a chromosome. So there are two chromatids here. So that's two halves. And then we have four telomeres, which are these leg bits here. Then the centromere is where the chromatids are attached to each other or linked. And so this is basically just a condensed form of DNA. Okay, so what happens next? So in mitosis, we have our second phase, which is metaphase. Metaphase is where the chromosomes line up in the middle. You can see here they are, single file, and they line up in the middle, and then the spindle fibers are going to attach to the chromosomes here at the centromeres. And that's going to be important because what's going to happen is the spindle fibers are going to pull these apart in our next phase, anaphase. So anaphase is where we pull apart those chromatids and we're pulling them to opposite ends of the cell. And you can see we're pulling one side this way, one side this way. We're pulling them towards these centrioles. And back here, this is just an electron microscope picture of a cell going through this. That's all this is. The green stuff is spindle fibers. The red is chromosomes. That's all you're looking at there. Okay, so now we have our next phase, telophase. And telophase is where we start to form new nuclei. So the nuclear membrane that we dissolved during prophase is now going to start developing around our chromosomes in our two halves of the cell. And we're going to start pulling apart here. And you can see the cells are starting to pull apart into separate cells. And while the nuclear membrane is forming, the chromosomes are going to uncoil into just regular strands of DNA. And then we have cytokinesis. Cytokinesis is where we're dividing the cytoplasm. That's what that word actually means. It means to divide the cytoplasm. And so the cell membrane pinches apart in animal cells and actually divides the cytoplasm that way, making two separate cells. In plant cells, cytokinesis works a little bit differently. Instead of pinching apart, what we have to do is separate the cells with a cell plate, and that is basically a segment of cell wall that kind of starts developing between the two cells in order to fully separate them. And so this is just showing you some other pictures. You can actually see here that cell membrane starting to kind of pinch in so that the cells can pull apart. And here you can see that cell plate starting to form here in our cell. So cell division becomes really important when we start looking at multicellular organisms. Multicellular organisms are things like humans and dogs and cats, things that have more than one cell. And because we have more than one cell, our cells have to do different jobs. And so we've studied a lot about how cells become different types. How do we get red blood cells? How do we get muscle cells? How do we get neurons? How do we get skin cells? And what we found is that as certain cells divide, they can actually turn on certain genes and become certain types of cells. And those cells that can divide and become different types are called stem cells. Stem cells can become specialized cells, or when they divide, they can continue to be stem cells. And we're trying to figure out how stem cells have this ability. Because once a specialized cell is formed, it can only ever make that type of cell. So, like, take a skin cell here. A skin cell, if it divides, it's just making more skin cells. It can't then become a neuron. And it would be kind of useful if we could turn on this ability and generate new cells, because that might help with some age-related diseases, or we might be able to turn on cells that have differentiated incorrectly and get them to go down the right path and become the right type of specialized cell. Now, we've talked about how cells reproduce themselves. What about how they die? So a cell's preferred form of death is called apoptosis. This isn't the only way they can die. This is just what we'd like to see happen. Cells can be eaten by other cells, or they can explode. Like in the case of like viruses infecting cells, they will actually inject their DNA. That cell will now become a virus factory, and then it will fill up with virus until it just explodes, releasing a whole bunch of virus to the surrounding cells. But apoptosis is different. Apoptosis is a controlled form of cell death. It's a controlled way for the cell to die, and it's sometimes referred to as cell suicide, but we're going to refer to it just simply as cell death. So this is controlled by a feedback loop. Basically, you have your body telling cells when they get too old to die. And as the cells are younger, they make a chemical that says, no, we're okay, we don't need to die yet. And then when they get too old, they're not able to make that. So they start going through this process where basically the cytoskeleton collapses and the cell membrane collapses in around the cell parts, and they form these bubbles of cell parts that they can give to other cells, and those can be absorbed by the surrounding cells, so they get recycled. So this is going to be just our quick overview. We're finished for now. We'll talk more in class.